So these are the wheat varieties that are entered into the experiment. In 2013, we had PBW 343. It is a susceptible check. The seedling infection type that we would read is a 3. Francolin is, we guessed it has 1 to 2 minor genes. The seedling reaction is a 3++. Danda, we think, may have more minor genes, maybe 2 or 3. The seedling reaction is a 3+. Kingbird is near immune response. May have as many as 4 or 5, possibly more genes. The seedling reaction is a 3++ or 4. So we're interested in these 3 adult plant resistant varieties and their response to fungicide protection. But we're also interested in knowing what the difference is, or a comparison of minor gene or APR response, APR protection, to a single gene that is a seedling gene. So the seedling response here is a 2 or 2+, so it is a seedling all-stage gene. In 2014, we had the same PBW 343 susceptible check, and then the 3 APR varieties. In 2013, we had a race shift, and so this variety here, Digaloo, became susceptible to the virulent pathotype TK-TTF. And so we could no longer use Digaloo as our all-stage resistant variety. And so we changed to Eagle 10, which is protected by SR catbird, and it gave a seedling response of 2 and 2+. And this is what kingbird looks like in the field. I don't know, how many of you have heard of kingbird, seen kingbird? Anybody? Yeah. Very resistant as compared to the susceptible check. This is the what the field plot layout would look like for a split plot arrangement of treatments in a randomized complete block. So here is the whole units, and they would be randomized in Rep 1, and they would be randomized differently, of course, in Rep 2 and Rep 3, and then the subunits, either with a fungicide protection or without a fungicide treatment applied, and these would be randomized within each whole unit. So if you'll count them, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 times 3 is 30 experimental units. Minus 1 is 29 degrees of freedom. So we're going to allocate the sources of variation into those 6 sources of variation in the analysis of variance table. Whole units or varieties randomized within complete blocks. Subunits, the fungicides randomized within the whole unit or within the variety.